Hello everyone. Today we are going to study a very important topic. Well, mostly the examiner doesn't ask this topic for your university exams, but they might ask for internal. But this topic is really important for you to study as a basis for other topic. If you want to study some disease like malaria, you need to know this topic for you to understand what's going on. Not malaria, but any disease, any bacterial disease. So, before we start this topic, I would like to encourage you all to study every topic as you are understanding why we are studying this topic. First, start with why you are studying any topic for any reason, right? Because today, as you're becoming a doctor, every patient has internet, right? They will ask you questions that will really confuse you if you don't understand the importance of every single topic that you're studying. Like for example, a patient might ask why you are giving multiple drugs for hypertension or diabetes. Yeah, you have studied that you need to give multi-drug therapy for any disease, tuberculosis or anything. They will ask you why you are giving this disease, this drugs for this. You're giving me four drugs for one disease. Why is that? But oh, you have only studied that you need to give 6 month HRZE therapy for tuberculosis. You have not studied why you are giving these four drugs. So if you don't understand the importance of each topic, it will really affect you in your clinical practice. So every time I teach you any topic, I help you understand why. Like in Robert's postulate in my previous video, I explained why we are studying these postulates. Also, when we are studying sterilization, disinfection, I explained you why that you need to know sterilization and disinfection as a basis for understanding any topic in any book. You are reading a book and you don't know the difference between sterilization and disinfection, it will affect you a lot. That's why I always encourage you all to understand why we are studying this topic. In bacterial growth curve, which is our topic of the day, I will explain why we are studying this topic as we go further. So it will help you understand the topic better and remember it for a long time. Let's start. Obviously, by the name, you can understand it's a curve, or to be more specific, it's a graph. And as every graph in this world, there is two axes, which is y and x axis. In this axis, the y represents the number of viable cells present in a media, in a liquid media, and x commonly, as you know, refers to as time. So why you are studying this curve? So basically, if you inoculate any bacteria in a suitable or in a liquid culture medium and incubate for a long time, it will show this curve. It will always show this form of curve. But there is a very important line here. It is suitable liquid media or favorable conditions. So why we are understanding this favorable conditions? So we can know what are the favorable conditions of a bacteria, and we can use these favorable conditions against them. If there is a bacteria which has a longer time for metabolizing and becoming bigger, we can try to affect that phase, which is the lag phase. We can try to affect that lag phase by using the drugs we need. So this curve is really important for you to understand why a bacteria shows this curve under favorable conditions. And if let's say I put a certain bacteria A in a liquid medium L, my temperature is X, my nutrition value is Y, all the micronutrients are A, B, C, D, E, F, everything, and the curve is not this curve, which is this curve. more specifically known as a sigmoid curve, but this is not particular sigmoid, but it looks like a sigmoid curve. So when if it's not showing sigmoid curve, we can try to change the favorable conditions a little to find what's the sigmoid curve, what's the favorable condition when it shows a sigmoid curve. When it shows the sigmoid curve, okay, we will know that this is now the bacteria's favorable condition. So that's why we always and always need to know what are the favorable conditions of a bacteria, and to identify what are the favorable conditions, we use this curve. So the definition is as simple: when a bacteria is inoculated in a suitable liquid culture media and incubated for a long time could be anything, incubation of which different bacteria are different, its growth follows a definitive course. When the bacterial count is plotted against the time, the graph is known as bacterial growth curve. This is simple, this is not rocket science here, but we will understand the phases of this bacterial growth curve. First, the lag phase. In the lag phase, which is the period between inoculation and the beginning of the multiplication of bacteria. So at the beginning, I inoculated my bacteria here, and the timer starts, and in this phase the bacteria is not actually multiplying. As you have studied in cell division in your 11th class, the bacteria is going under S phase. It is preparing for the replication phase. Right now it's metabolically active, and it is preparing for the M phase, which is if you study cell cycle you will know. So it is currently under interphase, as you know. Interphase is the period between two multiplications. So it use the time to build up enzymes and metabolites. It increases in size, reaches maximum size by the end of this phase. It increases its cytoplasm. Then comes the log phase. In this phase, the bacteria divides exponentially. You might think, why exponentially? Like, it can just divide, right? Why exponentially it divides? The answer is because of high amount of nutrients and minerals that are provided to the bacteria. Right now, the resources are abundant. That's why the population explodes and it divides exponentially. So the log phase is basically the exponential rise in the bacterial number, right? This is the rise in the bacterial number of cells, the viable cells. Now comes the stationary phase. What is the stationary phase? As the bacteria divides, it gets old, and the growth ceases almost completely during this phase. And why it does? Because of the exhaustion of nutrients, accumulation of toxic products and autolytic enzymes. Because the medium doesn't have not much resources, the bacteria ceases to divide and it starts dying. In this phase, the number of progeny cells are just enough to replace the number of cells that have died. So in this phase, many cells are dying. That's why the total count increases, but the stationary phase shows only viable count. That's why I said in the bacterial growth curve, the viable count of bacteria is important, right? The viable count, not the dead. We only need the living cells. But now, why this stationary phase is important is because bacteria becomes gram variable and cannot be stained uniformly. If you know what is the gram staining, I hope I can teach you in future. But in a gram stain, the bacterial cell contains different amount of cytoplasm and different amount of nucleic material, nucleus. That's why it becomes very difficult for the bacteria to be stained uniformly. There are many bacteria which will be stained heavily, and while other bacteria will be stained very less because they have already died. In this phase, when there is exhaustion of nutrients, more storage granules are formed so that they can prepare for prepare for the depletion of resources, and sporulation also occurs because it is an unfavorable environment now. It was favorable when the bacteria started to divide, but now it has become unfavorable. The bacteria also produce exotoxins, antibiotics, and bacteriocins. Now comes decline phase because there is so much accumulation of toxic materials and exhaustion of nutrients. The conditions have become unfavorable, and now the reduction in viable count can be seen, but not in total count because the total count contains both live and dead bacteria. Now there comes very important topic, which is factors affecting the growth of the bacteria. Why we need to know this is because we we are finding the favorable conditions, so we need to understand the what we can say the major factors that are involved, so we can understand it as grossly as we can, right? First, the major factor is oxygen. The oxygen, which is the bacteria which uses oxygen, are the bacteria which are known as obligate aerobes. Now I would like to teach this topic in very slow and complete manner. First, oxygen requirement. Any bacteria that cannot absolutely grow without oxygen, it needs oxygen no matter what, are obligate aerobes. They can only do aerobic respiration. They need they need oxygen no matter what. So they can only grow in presence of oxygen, and the examples of that are Pseudomonas, Microbacterium tuberculosis, and Bacillus. If the oxygen requirement reduces, then some aerobic bacteria need to do anaerobic respiration. They are called facultative anaerobes. They are aerobes, but under certain unfavorable conditions or under certain facility, they need to do anaerobic respiration. So they are aerobes that can grow anaerobically if needed. Most pathogenic bacteria comes under this, like E. coli and Staphylococcus aureus. After that comes the facultative aerobes. Now they are exact opposite of facultative anaerobes. They are anaerobic bacteria, but they can grow sometimes in oxygen medium, which is example of lactobacillus. Then comes microaerophilic bacteria. These are the bacteria. Now we are reducing the oxygen. You see, the oxygen requirement is reducing slowly and slowly. The microaerophilic bacteria requires some amount of oxygen present for them to grow, which is Campylobacter, Helicobacter, and Microbacterium bovis. Then comes the obligate anaerobes. They need absence of oxygen no matter what for them to grow. They need the absence of oxygen. Oxygen is lethal to them. They cannot grow with them because in the presence of oxygen, which is Clostridium. Now I would like to teach a very side topic in this. You can skip this part. This topic might not help you, but it will help you in pathology to understand a certain topic, which is gas gangrene. As you know, gas gangrene is caused by Clostridium, a certain species of Clostridium. Now my question is, why does the gas molecules or gas bubbles are formed in anaerobe but not in aerobe? The gas bubbles contain CO2, but both anaerobe and aerobe bacteria cause the formation of CO2, right? In both aerobic and anaerobic respiration, the final product is CO2 only. So why does the gas molecule only forms in anaerobe bacteria but not in aerobe bacteria? Well, the answer is actually very simple. Because the anaerobic respiration only forms lower 8 ATP for the whole digestion of one molecule, for producing 6 CO2 molecules, only 8 ATP is formed. Therefore, they need to destroy more and more cells so that they can get their certain requirement of X amount of ATP. Once they get their X amount of ATP, the production of CO2 becomes very high, because the anaerobic respiration only forms 8 ATP, but they need a lot more. But for 8 ATP, they produce 6 amount of CO2. But while in anaerobic, while the aerobic, they form 32 for the formation of 6 CO2. They get 32 more. So they need to destroy 4 molecules less, or 3 molecules less, for getting their energy. That's why the bubble CO2 formed in anaerobic respiration but not in aerobic respiration, or anaerobacteria but not in aerobacteria. The final category of oxygen is air-tolerant anaerobe. These bacteria can tolerate oxygen for some time, but they do not use it metabolically. Example is Clostridium histolyticum. This is a very simple topic right now. The oxygen one is the most important one. After that, I think you already know the carbon dioxide. Certain bacteria requires the presence of certain amount of carbon dioxide, which is 5 to 10, for growth. They are known as the capnophilic bacteria. They are called Brucellus abortus and Streptococcus pneumoniae. Then comes the temperature. Now most bacteria can grow around temperature of 37 degrees Celsius, which is the human body temperature. But there are certain exceptions. First, psychrophytes. They grow below 20 degree. They are called Pseudomonas. Not Pseudomonas. Pseudomonas is the example. They are not called Pseudomonas. The Pseudomonas bacteria is the example of psychrophytes. Then comes the mesophile, which grows within a temperature of 25 degree. Most pathogenic bacteria comes here. Obviously, 37 is their ideal condition, but they can grow even in 25 degrees Celsius. Then comes the thermophiles, that can grow above 100 degrees Celsius, which is Geobacillus steatothermophilus. Now, if you remember, I told you this bacteria act as a control for autoclave, because autoclave works above 100 degrees Celsius. They need the control of, or they need the presence of this to see if this bacteria is growing or not. If this bacteria is growing, temperature does not have reached 100 degrees Celsius, right? So they need presence of thermophiles, which is Geobacillus steatothermophilus. Then comes the pH. Now, pH for most bacteria, they grow on the neutral pH, which is 7.2 to 7.6. If you know, this is also within the range of blood. Blood is also the range of this pH. But the bacteria that can grow under below pH 4 is lactobacilli. Lactobacilli can grow below 4. They require an acidic medium for them to grow. While there are other bacteria, which is the pathogenic bacteria, which is Vibrio cholerae, that can grow in alkaline pH, which is 8.2 to 8.9. Now comes light. Now bacteria grow well in dark, obviously, except phototrophs. Why? Because they are sensitive to ultraviolet rays. We as human have some amount of resistance, but bacteria does not. That's why they are sensitive and they grow well in dark. But some bacteria, like photochromogenic microbacterium, they produce pigments on exposure to light so that they can protect themselves from ultraviolet rays. Now the last one is moisture. Simple, Trypanosoma pallidum, which causes syphilis and Neisseria gonorrhoeae. They die quickly after drying out. That's why they all grow in damp places or sexually transmitted. While others, like tuberculosis and Staphylococcus aureus, may survive drying out for several weeks. Drying in cold or vacuum, which is lyophilization, is used for preservation of microorganisms. This is the topic of today. It's a very simple topic, but it's also a very important topic. I hope you understand this topic very well, and I hope this topic helps you in all of your exams. Thank you very much. See you tomorrow.